Hello everyone, my name is Harsh Chauhan. I welcome you to this video. I am a teacher of plant biotechnology in the Department of Biotechnology at Indian Institute of Technology, Roorkee, and I will be discussing chapter number 6.2, which is related to plant cell culture and its application in biotechnology book of CBSE board for class 11th and 12th. So let's start our discussion. And what before starting actual discussion into plant cell culture techniques and its applications, I would like to discuss what do we mean by biotechnology per se. There can be many definitions of biotechnology, and one of the simplest definition which I found is that biotechnology is the use of biological processes, organisms, or systems to manufacture product intended to improve the quality of human life. When we talk about biological processes, that means the processes which we can modify in either inside the biological organisms or in a kind of a reactor system where biological processes can be carried out by certain organisms, say for example fermentation, which is carried out by either bacteria or yeasts. When we talk about organisms, that means we basically modify the whole organism, say for example plants, say for example microbes, to produce uh, like antibiotics or uh, products which has high food quality and so on. So if we also need to define plant biotechnology, then a simple definition for plant biotechnology would be that plant biotechnology is the use of plant-based biological processes, plants as an organisms or plant-based systems to manufacture products which are intended to improve the quality of human life. Plant biotechnology inherently has two components which are most of the time interlinked: plant cell culture, cell and tissue culture techniques, and genetic engineering and development of transgenic plants. So if we are talking about genetic engineering and transgenic plant, it is always involved in one form or the other plant tissue culture application and techniques. On the other hand, plant tissue culture alone is being done for various processes and improvement of plant-based products and quality. Also, when so much of technological developments are happening around us, we can ask ourselves, why do we need plant biotechnology? So there are many benefits for plant biotechnology. Some of them I have listed here. Say for example, plant biotechnology is helpful in farmers' benefits in one way or other ways. Say for example, increase in productivity or reduced cost by increasing yields. So if productivity increase, then it is directly related to farmers' benefits. If costs are reduced by minimizing the inputs, say for example in the form of fertilizers or in the form of tillage practices, we can make, if we are able to make crop plants which are better utilized, which can better utilize of provided applied fertilizers, then we can basically indirectly reduce the cost, which would result in the increase of farmers' income. Plant biotechnology is being used, and it has been used for many years uh, as of now, for making superior crop varieties which are resistant for insects as well as various bacterial or fungal diseases. These days, since environmental pollution is a concern for ourselves, environmental pollution is also a concern for our plants. Say, for example, air pollution, deprivation of water, depletion of water table, degradation of soil, increasing environmental temperature because of this phenomena of global climate change. So plant biotechnology is being used to make newer and better crop varieties for which has more tolerance towards these abiotic stresses. These are collectively called abiotic stresses, such as heat stress, drought stress, and environmental pollution. Plant biotechnology is also being used to make plant products which has better processor benefits. Say for example, plant biotechnology is being used in making sugarcane varieties which are softer but at the same time has more sugar content. There are consumer benefits also which are linked to plant biotechnology practices, say, such as increased protein, vitamins, and other nutrients in uh, our food crops. Say, for example, more protein in wheat, or more protein in rice, or more vitamins and beta carotenes in wheat or rice, and also healthier oils. That means plant biotechnology is being used to make oil crops such as brassica and uh, rapeseed, which has more polyunsaturated fatty acids, or popularly called as PUFA, or omega fatty acids, which has more omega fatty acids. Plant biotechnology based crop plants are being developed. And apart from all these, which are direct linked to the consumer benefits or farmers benefits, there are indirect benefits also for by applying plant biotechnology based practices, which basically benefits our environment indirectly. Say for example, if we need to put less fertilizers or less insecticides or less pesticides to grow our crop plants in farmers field, that means we are indirectly benefiting our environment also, because if the use of these harmful chemicals is reduced, then environmental got benefit. It also, plant biotechnology also helps in conserve of soil and energy. It helps in reducing greenhouse gases, minimize the use of toxic herbicides and insecticides, and all these things collectively results in the increase of soil fertility and natural resources. So these are some of the benefits I could list related to plant biotechnology, but there are many more things which can be done with the help of plant biotechnology. So when we say plant biotechnology, if you see this picture, uh, it's a simplified version of how plant tissue culture is done, which is also which is actually one of the unit of plant biotechnology. So plant tissue culture in the last 30-40 years has seen a long development in a way that it is technically now possible to tissue culture each and every plant, provided that we identify three crucial elements, such as which part of a plant should be taken for plant tissue culture, what type of growth and nutrition media is provided to that part of plant so that it can be used in plant tissue culture, and third is the growth conditions. What type of growth conditions, say for example light conditions, temperature conditions, humidity, and other things are provided so that the chosen part or chosen plant part on a chosen media gives us the intended product in plant tissue culture practices. In general, plant tissue culture refers to taking any part of a plant, say for example root, shoot, stem, leaf bud, flower, or part of even flowers such as anthers, embryos, embryo sacs, ovary, to develop a complete plant or even a group of plants. That means many plants can be developed from one single source of a plant. When we use this plant part, it is collectively called as explant. So when I listed roots, stems, leaf buds, all these are collectively called as explant. Second one is which I used the term nutrient media. Nutrient media, uh, there can be many types of nutrient media. Like humans, plants also need nutrition, and I will be discussing in subsequent slides what do we mean by nutrient media as well as growth conditions. So everything related to plant part, nutrient media, and growth conditions will be discussed in subsequent slides. Now I have divided this chapter into certain kind of subsections which are related to introduction and the history of plant cell culture and genetic engineering. And then I will discuss with you what minimum requirements we need and what are the minimum tissue culture practices we do in terms of laboratory setup, cell and tissue culture techniques, gene transfer methods in plant, formation of transgenic plants with beneficial traits. I will discuss certain examples related to that. And in the last, I will discuss biosafety in plant genetic engineering or in plant tissue culture, and how these things are being used in day-to-day life around the globe. So when we say plant tissue culture, as I have just shown in that picture, plant tissue culture is a game of plant cell tissue type, collectively called as explant, which can be of leaf, shoot, node, bud, pollen, anther, or in another way, any plant part or group of cells. Then we need to have an important nutrient media which is a source of different type of nutrients. Say for example, as humans, we need different type of nutrients. We need carbohydrates, we need proteins, we need vitamins, we need uh, uh, minerals for a proper growth and development. Same way, plant also need all these things for their proper growth and development. We also need hormones, plants also need hormones. And then specific culture conditions are provided, followed by the hardening or the transfer of regenerated plants to soil, and then we carry out the test for our intended purpose for which we have done the plant tissue culture. So before discussing the tissue culture practices and applications, let's discuss a little history of plant tissue culture. So if we go back uh, to see how plant tissue culture started, the origin is basically uh, can be seen in early 19th century, uh, related to tissue culture, and which applies to both animals as well as plants. The famous theory, cell theory, proposed by Theodor Schwann and Matthias Jakob Schleiden in 1838, or also famously listed in all the textbooks as Schleiden and Schwann cell theory. What they have postulated in this theory, that all living organisms are composed of one or more cells. So either if we are talking about microbes, they are single cell organisms, and if we are talking about eukaryotes, then they are multiple cell and multi-organ organisms. The cell is the most basic unit of life. All cells arise from pre-existing cells. That cells cannot be artificially made. Well, there is an exception now, but cells arise from pre-existing cells only, either by means of cell division or by formation of embryos and development of whole organisms. And the activity of an organism depend on the total activity of independent cells. That means if there is a multi-organ uh, organisms, say for example humans, there are organs, then organs are made up of tissues, and tissues are made up of specialized cells only. So ultimately the activity of an organism as a whole is dependent on the total activity of the cells. This is the famous cell theory proposed by Schleiden and Schwann, and this is basically the starting point for tissue culture for both plants as well as animals. In case of plants, there was a big development in 1846 when Julius von Schecks and Knopp basically for the first time they grow plants without soil. So what they took, they provide a solution of calcium nitrate, I mean appropriate amount of calcium nitrate, magnesium sulfate, uh, potassium dihydrogen phosphate, and uh, ferric sulfate. So if you see here, I'm sure by now you are aware about what are the major uh, nutrients which we need as far as elements are concerned. These are calcium, magnesium, sulfur, potassium, phosphorus, and iron. So if you see in this solution, all these 6 7 elements are provided, which uh, in the different form of salts, such as calcium and nitrogen is provided by calcium nitrate, magnesium and sulfur is provided by magnesium sulfate, potassium and phosphorus is provided by potassium dihydrogen phosphate, and iron and sulfur is again provided by ferrous sulfate. So what they showed at that time, that even if soil is not there and you have a solution of these basic salts, plants can grow. Later on, as far as plant biotechnology is concerned, plant tissue culture is concerned, a huge development happened in early 20th century by Gottlieb Haberlandt. He's an Austrian botanist working in Germany, and he was able, by taking idea of these nutrient solution, he was able to basically grow leaf mesophyll cells for a few days. Although he was not able to basically get them divided and make new cells, but he was able to grow mesophyll cells for a few days. That means without providing actual plant part, isolated cells were able to keep living on a provided basic nutrient solution comprising of the salts which we have listed just in the previous slide. And he proposed that provided a suitable nutrient solution and growth hormones. Now, this was a huge development. Provided the nutrients and growth hormones, we can induce cell division in plant cell culture techniques. And later on, he actually got dividing cells when he used embryo sac fluids for the supply of plant hormones. And he also postulated that artificial embryos can be cultivated from vegetative cells. So this is one of the thing which proven right in next 30-40 years, and which is being used these days. And the embryos which we develop from vegetative cells are called somatic embryo. I will discuss somatic embryo either in the subsequent slides or in the next video when I will be discussing the actual applications of plant tissue culture. And then Professor Philip R. White was the first one who basically got the dividing cells from root tips of tomato in the nutrient solution. And he found that if we use root tips or shoot tips, which is virus free, then the whatever cells the mass they develop into also has also is virus free, and the cell division for plant tissue culture can be induced by providing vitamins and certain growth hormones. He was the one who gave the famous concept of plant cell totipotency. So totipotency is a word derived from Latin word totipotentia, which literally means ability of all things. So plant totipotency is actually can be defined as the ability of a single plant cell to divide and produce all of the differentiated cell in an organism. In general, taking a plant cell, isolate it, provided right nutrition, conditions, and growth hormones, it can be developed into a whole plant or a bunch of whole plant, maybe a few hundred plants. This is called plant concept of plant cell totipotency. In general, in natural condition, it also happens all the time in the form of spores and zygotes. Spores and zygotes are unicellular initially, and then they develop into embryo or the whole plant. This was a huge development, and then for next 30-40 years many sequential development happens. One of the I have listed some of the important, and one of the most important development was the isolation of first plant hormone, that is indole acetic acid. It was isolated in year 1934 by Wendt and Thiemann, and then when it was included in plant tissue cultures by them with having this nutrient solution of those salts and certain vitamins and IAA is added, they were able to grow unorganized mass of cells called callus, which is basically result of rapid cell division by using either root tips or either shoot tips of carrot, tobacco, and willow. In 1941, coconut milk was used for the same purpose because coconut milk contains almost all types of nutrients as well as growth hormones. In 1949, Limasse and Cornau, they demonstrated that plants developed through micropreparation are virus-free if the source of plant cell culture is taken from virus-free shoot tip or virus-free root tip. A huge development happened in our country also when in Delhi University, Professor P. Maheshwari and Professor Guha in 1964, they developed a haploid plant by culturing by culturing anthers of, I mean the plant part of flower anthers on a suitable media. Anthers was of plant species Datura, and it was first instance of developing a haploid plant from plant tissue culture techniques. What are haploid plant and what are its application? This will I will also discuss in subsequent slides or in another video, following video. Then a development which basically changed everything happened in 1965 when Indra K. Vasil and Hibble and culture and isolated one single cell into a whole plant, and that was the experimental proof of concept of plant cell totipotency. Following that, first commercial cultivars were developed in canola and barley by plant cell tissue culture, and these days plant cell tissue culture is being used for development of improved varieties in almost every type of crop plant or horticulture plant or flower plant. Say for example, orchids are being produced by plant tissue culture, ornamental plants are being produced by plant tissue culture, newer flower varieties are being developed by plant tissue culture. And the second component, which I said related to plant biotechnology genetic engineering, it has been known that certain bacteria, a bacteria called agrobacterium, is able to basically change the genetic composition of target plant. But in laboratory, agrobacterium was first used in 1983. The paper was published in one of the most reputed journal, Nature, and first transgenic plant was developed by using agrobacterium in tobacco. After that, it was kind of an exponential growth as far as plant tissue culture is concerned, and last 2 decades showed tremendous growth in plant cell culture techniques and its application. These days, all in our day-to-day life, we encounter products which are resultant of plant cell tissue culture methods and its applications. So what is there in plant tissue culture which basically is so useful? Why people do plant tissue culture, or why we need plant tissue culture as far as the practice is concerned, apart from its applications? There are many possibilities. Some of them I listed here, that a single explant can be multiplied into several thousand plants in less than a year. So if we established a proper plant tissue culture method of our crop of choice, say for example sugarcane, so there are varieties of sugarcane which are developed by plant tissue culture. These varieties are either resistant to certain kind of pests or insects, or they are resistant, they use less water. So these varieties, if we try to multiply them in soil and in the field, it takes 1 whole year or maybe 2 years to get enough seed material or enough planting material. On the other hand, millions and millions of small saplings can be produced for important sugarcane varieties within a year in a plant cell tissue culture based laboratory. Second benefits, if we take an explant from the mother plant, it do not destroy the whole plant. We only take a plant part. Say for example, we take either shoot tip or root tip. The mother plant is always there to provide more material. We need not to kill or to cut down the whole plant. So the source is always there. We only take a part from the soil from the source which can be developed into a whole plant, thousands of plants. Another benefit is if we have established a particular system for a crop of choice, then we can basically keep on producing our plants for maybe many years, 10, 20, 30 years, without disturbing the original source of the X plant. Another benefit is that plants so produced by plant tissue culture are virus free if we have taken plants explant from a healthy plant. Tissue culturally grown plant can be screened faster than the plants which we grow in soil or in the open fields. And one of the important thing that tissue culturally grown plants are basically exact replica of the source plant. They are true to type. That means there is no change happens. Normally, there is no genetic change happens if we are not inducing the change from outside. In general, no change happens in tissue culture. And third is because of they have higher quality, they have more easy and high export potential to foreign countries. For example, most of the orchids which we see these days in our market, they originate from one single country, that is Thailand. So if we can establish a reliable, reproducible, and economical method for plant tissue culture of an important cultivar, then since the resultant plants have better quality, they can be characterized easily, and they are disease free, they have very high acceptance in the market, as well as they have high export potential which can earn money to the companies or the person involved in plant tissue culture. Now, what are the basic minimum requirements for plant tissue culture? I could list three basic requirements, which one is related to laboratory organization, second is related to suitable nutrient media, and third related to identification of a suitable explant depending upon what type of crop plants we are interested in. So when it comes to laboratory setup, the minimum requirement for a laboratory setup is washing and media room. That means where we do day-to-day washing as well as preparation of nutrient media. Second is inoculation room. That means where exactly we do the plant tissue culture practices. Third one is after doing inoculation or doing the plant tissue culture practice, where we keep our tissues or cells to grow or to develop or to undergo further growth and development so that they can make whole plant. And the last one is if we are dealing with transgenic plants, then we also need a transgenic containment facility. Out of these components, washing and media room and inoculation can be in a same big room into two different corners, and culture room can be a third extension to the same facility, as I will show you in subsequent slide. Normally, transgenic containment facility is outside than the actual plant tissue culture laboratory. So, I mean, for your clarity, if you see in this picture, this is a media room which we use here at IIT Roorkee for plant tissue culture purposes. It's a partition of a big room, and there are basic instruments are here. There is a washing area, and then there is a working bench, and then there is another bench housing basic instruments such as balance, magnetic stirrers, pH meters, and in one of the corner there is this big autoclave which is used for sterilization of instruments as well as nutrient media. So apart from these, many other things are required such as microwave or other oven, refrigerators for keeping expensive chemicals or chemicals which needs to be kept at lower temperatures, lab carts and trays, autoclaves, and certain other things. So this is how a media room looks like. Then the second part is inoculation room. I will cover it quickly. The one of the most important thing as far as plant tissue culture is concerned is this chamber which is called a laminar airflow hood. Basically, it is a chamber where we keep where even the air is sterile. it composed of an air pump, and the pump is connected with HEPA filters. HEPA means high efficiency particulate air. So if you see this chamber, the air suction is created in the upper chamber, and then the air is recirculated from this side, the white side, on this hood or the shelf, and whatever comes from these white filters, which are HEPA filters, every particle is basically kept behind this, and a pure and sterile air is let to enter this transparent chamber. So apart from getting sterile explant, sterile nutrient media, and sterile apparatus which are used for plant tissue culture, sterile air is created with the help of HEPA filters in this laminar airflow hood, so that no contamination or microbes can infect our uh, plant tissue culture practices during development of tissue culture grown plants. Apart from HEPA filter, depending upon what type of plant tissue culture one lab is involved or a person is involved, there can be other instruments which are required. Say, for example, dissecting microscopes, tissue culture instruments such as scissors, forceps, and other things, flame sterilizer to instantly sterilize the minor instruments such as scalpels, blades, scissors, and tweezers, and other consumables, say, for example, tissue papers, uh, things related to discarding the waste, and some other instruments such as orbital shaker if the plant cell culture involves some kind of shaking of either tissue culture media or any other sterilization processes. Then the third important part is the culture room. So if you remember in the first slide, I told you that plant tissue culture is a game of three things: explant, nutrient media, and the growth conditions. So culture room is the place where we provide proper growth condition in terms of three major things. First is humidity, second is intensity as well as duration of light, and third is related to uh, temperature. And uh, most of the time we also try to mimic a day-night rhythm for our plant tissue culture practices. If you see here, these round plates are called Petri plates, and you can see that there are certain plant tissues are kept there. So normally we provide a specific intensity of light for a specific duration, and the most popular duration is 16-hour light and 8-hour night conditions provided by artificial means. The artificial lights, either LED lamps or these day-to-day light which we use in our homes, these tube lights. Afterwards, if you see here, the plants have been regenerated and they are kept in tubes. And from these tubes, the plants, after hardening and rooting, they goes into soil, potted soil, or directly into the fields for further development. And if we are dealing with transgenic production of transgenic, then we also need one transgenic containment facility. It is a greenhouse. You can see it's the picture of a greenhouse. This is the control panel. Again, this control panel is used for controlling light intensity, temperature, light duration, humidity. And you can see these are the transgenic plants. These are transgenic rice plants which are being grown in transgenic greenhouse. Other things which are there in transgenic greenhouse is a watering facility, a small storage and washing facility, and maybe some certain racks to keep pots as well as fertilizers or soil. Before we start actual plant tissue culture, some common basic personal training is required. These trainings are related to handling of the instrument. One cannot just go and start using these delicate instrument. There can be chances of accident. So a bare minimum training is required related to handling of instrument, how these instruments are used. The person who intend to use plant tissue culture laboratory needs to learn the instrument kept there, how, and how they are used. One of the important thing is to basically control accidental things, such as if there is an accidental fire, how to control a fire. I mean, it depends what is the source of fire. If it is ethanol or alcohol led fire, that means it needs to be doused by water. But if there is an electric fire, then we cannot use water, and there is the other means, so which is to cut the source of electricity. So basic personal training is required in terms of these things. Also, the training is required for handling hazardous chemicals, and how not everything can be washed the same way. How to wash our instruments, or glasswares or plasticwares. Training is also required for how to handle sterilization procedures. Say, for example, how to handle an autoclave, which is kind of a big pressure pot which we use in cooking in our kitchens. It's kind of a big pressure cooker, and a basic knowledge of first aid is also required to work in a plant tissue culture laboratory. Now, after, if we have instruments and if we have a plant tissue culture lab, how to do plant tissue culture, or how to grow our plants? Plants need food. For tissue culture also, our explants need food. So in plant tissue culture, mostly media are divided into two types: undefined or complex media, chemical or defined media. As the name suggests, undefined and complex media are those media for which we exactly do not know what is their ingredients. Say, for example, coconut milk. Coconut milk is also called as a complete foods, like we call milk is a complete food which has everything. In case of plants, coconut milk is is called as a complete food which can be used for plant tissue culture, but we exactly do not know what is the composition of coconut milk. On top of it, coconut from a one plant may have a different composition from coconut from a different plant, or maybe from a different region. It also depends upon the growth conditions of coconut trees. So this is collectively called as undefined or complex media. This is not much preferred as far as plant tissue culture is concerned, because we cannot replicate the exact conditions every time in the laboratory. The other type of media is chemical or defined media, which is a preferred type of media, because we know the exact composition of different constituents of nutrient media, and it can be replicated each and every time when we prepare a chemical or a defined media. There are many chemical and defined media developed. Uh, I will discuss some of the examples in subsequent slides. The major components of a chemical or defined media are inorganic salts, organic compounds, vitamins, growth regulators or hormones, and gelling agent. Let's discuss them one by one. So inorganic salts are again divided into two major categories: macroelements and microelements. Macroelements are those which are required in millimolar concentrations. These are called macroelements because they are required into higher amounts. Microelements are those which are required into much less concentrations, like in micromolar concentration, 10 to the power minus 6 uh, concentration. Millimolar is 10 to the power minus 3 concentration. So macroelements are in general which are required in zero point 0.5 millimolar or more, and these are 6: nitrogen, potassium, phosphorus, calcium, sulfur, and magnesium. Microelements which are required in micromolar concentrations are iron, manganese, boron, copper, zinc, iodine, molybdenum, and cobalt. In defined medias, it has been defined that which composition is required into which concentration. We will discuss it in subsequent slides. Organic ingredients are related to carbohydrate, that means a source of energy. Say, for example, humans needs we get energy for our functions from sugars, carbohydrates. Same way, plant cells also need energy, and they also get their energy in the form of carbohydrates. The most popular carbohydrates which we provide as an energy source in plant tissue culture is these mono or disaccharides, glucose, fructose, and sucrose. Like humans, plant also need vitamins. If Since in tissue culture they will not be doing photosynthesis and they are not producing their own vitamins, initially we need to provide vitamins in the growth media. Like animals, plant also need the same types of vitamin A, B, and say for example vitamin A, different vitamin B, and vitamin C. So in defined media we provide a defined concentration of vitamins, also vitamin B1, B3, and B6, and other vitamins. Generally, these vitamins are provided in a concentration of 1 milligram per liter, or even less. This concentration is enough. Then the one of the most important, or I would say most important ingredient of suitable plant growth media is growth regulators or plant hormones, because without providing growth hormones or growth regulators or hormones, the cells will not start dividing and developing into the whole plant, which is the basis for plant cell totipotency. And different plant hormone categories are auxins, cytokinins, gibberellic acids, abscisic acids, and these are the common ones, and others are colchicines, and sometimes charcoal is also used as a growth regulator. So auxins are basically responsible for cell division. Cytokinins are mostly responsible for cell differentiation. Gibberellic acids are mostly required for elongation of cells, and abscisic acid is mostly required for cell dormancy. So a, a defined composition and combination of these growth hormones are provided in growth media depending our purpose. And then last but not the least is the gelling agent and the pH of growth media. Most common and cheap gelling agent is most economical gelling agent is agar. Agar is basically a non-reactive uh, uh, polymer of originated from cellulose. It is made by certain C algae, and it's white in color, and it is completely non-reactive. It just provides the solid, it is just used for the solidification of our growth media. If we do not provide gelling agent, then the nutrient media remains in liquid form. When we provide gelling agent, then our nutrient media becomes solidified. And there are purer forms of agar which are basically sometimes used in plant tissue culture. They are called phyta agar or phyta gel or gelrite. And then there is these days a synthetic gelling agent is also used, which is called biogel P200. All these uh, chemicals are used for solidifying of our nutrient media. And pH is basically maintained around 5 to 6, and most preferred pH for plant cell tissue culture is slightly acidic, which is 5.8. So I'm sure you know what is acidic pH, what is basic pH. For better growth of any living organisms, or most of the living organisms including plant, a slightly acidic pH is better. Now, this table lists some of the popular uh, uh, media which are used for plant tissue culture. They are basically named on the basis of the people or the scientists who have developed them, like niche media, white media, Mauritian scope media, and so on. Here you can see that these are the ingredients which are used to make this defined media, and this is the concentration in which these these ingredients or these chemical compound are used to prepare these defined media. You can see here that top of them are macro elements, followed by microelements, followed by vitamins and uh, carbohydrate source here. So most of the time we use Murish and Skog, one of the most popular media, which has the exact concentration or exact composition as listed in this table. This is one of the most common media which we use. Now, after, how do we make a defined media in the lab? So we take deionized water and we add different components such as microelements, vitamins and carbohydrate source followed by gelling agent in a sequential manner. Normally we first add macroelements from a stocks which is made into 10 to either 10 to 50 times concentration, microelements, vitamin. So basically we add all these things in a sequential manner, and after that we adjust the pH to required and suggested pH. Most of the cases it is pH of 5.8, and then we add growth hormones. Now when we made this nutrient media is basically cake for every type of cell to grow. Say for example microbes, bacteria, fungi, they are everywhere. So before using this defined media for plant tissue culture, we need to sterilize this. That means we need to basically get rid of all the bacteria and microbes which are already present in our ingredients. And how we do this? This is listed as the maintenance of aseptic conditions in plant cell tissue culture, and there are three things which needs to be sterilized. The media, containers and small instruments needs to be sterilized. Then where we work, basically that area needs to be sterilized, that is the laminar flow hood as I just mentioned, and the part of plant which we are going to use for plant tissue culture, that also need to be sterilized. And if we work with our bare hands, our hands also needs to be surface sterilized. So what are the methods available to us? Most of the time the instruments and the media and the vessels, glasswares, all these things are sterilized by autoclave. As I told you, autoclave is just like a big pressure cooker. It creates a temperature of 121 degrees centigrade by using water steam and a pressure of 15 environmental pressure or 15 psi. That means under these harsh conditions, such a high temperature and such a high pressure, no living cell can survive. And whatever we put inside these autoclaves, they are of many types, vertical as well as horizontal. Whatever we put inside these pots, these big pots, gets sterilized for anything which cannot live on this high temperature as well as this high pressure. On the other hand, media, many of the time media cannot be sterilized by such high pressure because some of the ingredients inside the media such as vitamins and growth hormones, they are uh, they are destroyed, or their properties are basically destroyed at such a high temperature. In this case, media are sterilized by filter sterilization. Two types of filters are available. Bottle top filter, we put our media on top of this bottle top filter, we provide vacuum, and the sterilized media is collected in the bottle which is uh, at the lower part. Sometimes if we are not dealing with very big volumes, say for example if we need to sterilize only 5-10 ml or 20 mls of a nutrient media, or a composition of nutrient media, then these small filters are available which can be fit on plastic syringes, and by putting pressure from our fingers, and the whatever passes through these filters gets sterilized. When it comes to the area which we are going to use for plant tissue culture, this surface is sterilized by applying alcohol as well as ultraviolet lights for say for example 20 minutes. Whatever living material is here can be sterilized or killed with the combination of ethanol wash as well as UV lights. So this bench is cleaned by a combination of UV light as well as washing with ethanol, wiping with ethanol, and afterwards HEPA filter will provide sterile air when we start using this bench under normal conditions, when normal lights are on and UV lights are off. Another thing is how to sterilize the cells itself, which we are going to use for plant tissue culture. So there are many methods, and the most popular method is washing uh, our our explants or plant part with commercial bleach, which is sodium hypochlorite, as 1% to 4% solution, depending upon what type of explant we are using, can be used to sterilize. Just washing our plant parts with 1 to 4% solution of sodium hypochlorite, followed by washing with deionized water, autoclave deionized water, will sterilize, surface sterilize the explant which we are using. If in case our explant is very dirty, it is very old, or it is basically surrounded by soil and any other contaminant, then sometimes we also use mercury chloride for surface sterilization. But use of mercury chloride is not preferred because it is very toxic, and it also harms human beings. So most popular method of doing surface sterilization of explant is done by sodium hypochlorite only. Now, I I came to the end of this video, and I listed certain small questions which you should be able to answer after watching this video carefully. There can be many questions, by the way, but I have listed some of them which are related to what we have discussed in this video. Some of them are: what is a plant biotechnology, or what is biotechnology itself? You should be able to answer this question to draft a simple definition based upon this video. Uh, which biotech crops, or what are the benefits of biotech crop which we produce for plant tissue culture? What is the history of plant tissue culture? Which was the first hormone which was uh, discovered, plant hormone? Who provided the cell, famous cell theory, and what cell theory actually says? What do we mean by totipotency, and in terms of plant cell totipotency? You should be able to answer these questions. What do we mean by a tissue culture media? What are the major and minor ingredients of tissue culture media, and how we prepare a tissue culture media inside the lab? What are the major components of a plant tissue culture laboratory? What are the sections there? What are the bare minimum instruments we require? How we sterilize different things? Say, for example, how glasswares are sterilized, how nutrient media are sterilized, how explants are sterilized, and how the surface where we work during plant tissue culture is sterilized, and uh, how or minimum what basic type of training is required for working in plant tissue culture laboratory. In the next video, I will discuss with you the actual plant tissue culture uh, procedures and its application. Till then, you try to answer the questions which I listed in the previous slide. Thank you.